Hello class and good afternoon. This is Lindsey Kane and today I've made for you a pizza cell model, and I'm going to use this to help explain organelles through the use of this delicious looking pizza. Start off with a few basic structures just to help you understand where we're at. You can think of this crust as the plasma membrane, which is the outside of the cell. The sauce on here would be considered the cytoplasm, and all the organelles lie within there. And then the first organelle we're going to actually talk about is the nucleus. This in the very center is the nucleus, and it's considered the control center of the cell. It houses all the DNA as well as the nucleolus, this little structure right here, and the nucleolus is responsible for making ribosomes, which we'll discuss later. Surrounding that nucleus is a nuclear envelope, which is very similar to the plasma membrane. The second structure we will discuss is called the endoplasmic reticulum, or ER for short. It's composed of two different parts. A rough ER, which is this part right here that has the ribosomes embedded into it, and the smooth ER, which does not have any ribosomes on it. It's actually a series of continuous canals and channels called cisternae. So the rough ER, this part right here, has those ribosomes that were created in the nucleolus. Their job is to take coded DNA from the nucleus and then create peptides and proteins based on that code. The smooth endoplasmic reticulum does not have the ribosomes, and it functions to detoxify the cytoplasm. The next structure that we'll talk about, or next organelle, is right here, and that is the Golgi complex. That Golgi complex is composed of the same cisternae as the endoplasmic reticulum, only these are shorter and stacked. The Golgi complex actually takes unfinished peptides and proteins created in the endoplasmic reticulum and completes their synthesis. It also synthesizes carbohydrates. Once they are finished, it packages them and ships them into these little structures here, which are Golgi vesicles. And those Golgi vesicles will travel throughout the cell and the cytoplasm. The next structure we'll discuss is the lysosome. And lysosome is a type of Golgi apparatus. It's a type of Golgi vesicle. It is composed of interior is about 50 enzymes that are used to digest and break down different matter and waste within the cell. One of the things that it does is it breaks down bacteria and white blood cells. In addition, liver cells use it to release glucose into the bloodstream. The next one we'll discuss are these little guys right here called peroxisomes. They are very similar to lysosomes in that they detoxify the cytoplasm, and they do this by producing hydrogen peroxide, which they use to break down particles in the cytosol. The next organelle we'll discuss are these right here, and those are called mitochondria. And they are actually structures that are considered the powerhouse of the cell, and they're called the powerhouse because they produce ATP, which the body uses for energy. And if you see in here, we can imagine that those little things in there are kind of internal folds, and those are called cristae. And what these folds do is increase the surface area, which allows them to produce even more ATP. And then the final organelle we'll discuss are these right here, and those are called centrioles. And these are used in cell division. They're basically small rods composed of microtubules that help in organizing and orchestrating all the components that go into cell division to make sure that that process goes smoothly. And if you note, they're always in pairs of two, and they're always in a perpendicular fashion. And that completes our model of all the organelles. So I hope you have a great day, and we'll see each other soon.